Alright, here we are in Chapter 16, Biochemistry, that third part of our general organic and biochemistry portions of our fundamental chemistry course. And I know many of you may have interest in the health sciences, and obviously biochemistry is a critical aspect of the course for you, and all of us should have a better understanding of biochemistry. So hopefully this will be an exciting chapter for all. Looking specifically at our learning objectives for this chapter, we will be able to list the major parts of a cell and describe the function of each part. We'll name the primary source of energy for plants and three classes of substances that are the sources of energy for animals. We'll compare and contrast starch, glycogen, and cellulose. And we'll describe the fundamental structure of a fatty acid and of a fat. In addition, we'll classify fats as saturated, monounsaturated, or polyunsaturated. We'll draw the fundamental structure of an amino acid and show how amino acids combine to make proteins. We'll describe the four layers of protein structure and give an example of each. And we'll describe how enzymes work as catalysts. We'll also name the two types of nucleic acids and describe the function of each type. We'll explain complementary base pairing and describe how a copy of DNA is synthesized. We'll explain how mRNA is synthesized from DNA and how a protein is synthesized from messenger RNA. And we'll list important characteristics of the genetic code. And finally, we'll describe recombinant DNA technology and explain how it is used. We'll name some advantages of using biochemistry to create useful molecules. And we'll give examples of the use of biochemistry for energy production and other applications. So lots of things to accomplish in this chapter, and as a result, we'll get right to it. So defining biochemistry. Biochemistry is the chemistry of living things and life processes. Most institutions put biochemistry along with chemistry, although a few noteworthy examples, like Cornell up the street, has historically put the biochemistry in with the biology department. When I was just getting my own bachelor's degree in chemistry, it was just about the time that biochemistry was recognized as the fifth pillar of chemistry. So traditionally, up until around the year 2000, chemistry had four subdisciplines. You had inorganic chemistry, organic chemistry, analytical chemistry, and physical chemistry. And then right around the year 2000, biochemistry was officially recognized and embraced by chemists as the fifth pillar of chemistry. And indeed, a lot of the major discoveries in chemistry and the Nobel Prizes awarded in chemistry have been in the area of biochemistry. So it's an incredibly important area, and one that is clearly interdisciplinary among biologists and chemists, and biochemists sort of straddling the line there. And so it's an exciting chapter and a chance for us to get to see exactly what biochemistry is all about. So if biochemistry is the chemistry of life, then we'll take a look at the basic unit of a living organism, and that is the cell. Again, things get a little interesting here. Viruses sometimes are living, sometimes are not. They don't have the cell, they're subcellular. But keeping things neat and clean, we'll call the basic unit of life the cell. And therefore, if we examine the cell, we'll note that cells are enclosed in a cell membrane, and that membrane selectively regulates the passage of nutrients and waste. And so in addition to the cell membrane in all cells, plant cells also have a cell wall composed of cellulose. And one of our learning objectives was to distinguish between cellulose and starch, and we'll do so momentarily. Alright, going even more subcellular, the nucleus. The nucleus contains the genetic material that controls heredity, and it's located in the nucleus of the cell. Ribosomes, these small structures in the cells, are where protein synthesis occurs. Mitochondria are cell structures where energy production occurs for those of us organisms that are aerobic, like humans. And then chloroplasts are found only in plant cells, and This is where photosynthesis occurs within the chloroplasts. So we see here a diagram of a plant cell, and as we mentioned, we have some of those key features. In the nucleus, obviously, there's even substructures, the nucleolus, nuclear pore, chromatin, and the nuclear envelope. We see some ribosomes there by the nucleus. There's that cell wall we mentioned that plant cells have and animal cells don't. And then, of course, the plasma membrane that all cells have. A number of other subcellular components that, for time's sake, we won't go into, but I'm hoping you remember from your previous bio course, whether it was high school bio or some college bio. Not a prerequisite for our course, but hopefully you have had a little bit of biology, because we won't have a chance to go as deep into the biology. We're just sort of glossing over a lot of that so that we can get into the biochemistry. And here we see a picture of an animal cell, again, similar to the plant cell. We have all the nucleus and the subnuclear structures, but obviously, while we still have that plasma membrane, we're lacking the cell wall that the plant cell has. So we noticed in the previous image, the cell wall gives that rigid structure to the plant cell. The animal cell is more spherical here in this artistic rendering, and a lot less structurally fixed because of the lack of a cell wall. But we've got all those other subcellular structures, some of which we mentioned, some of which we haven't mentioned exactly, like the Golgi complex that hopefully you remember, and cell vesicles and centrioles, all those things that you should have had at some point in your prior education, but again, for sake of time, we won't deal with them directly. Obviously, the nucleus will be quite important to us as we talk about nucleic acids as part of the major macromolecules. All right, so energy. Energy is obviously important in our world today. We've been through various energy crises, but energy for biological systems is another area of quite a bit of interest. If we look at green plants, obviously green plants are critical to our survival. Animals can't make their own food. We have to rely on plants as a food source, and so it's the green plants that can take that energy from the sun and store it as chemical energy in glucose. We see that photosynthesis reaction here as we've seen it before in the course: 6 moles of carbon dioxide molecules plus 6 moles of water molecules with the sunlight proceeds to yield 1 mole of glucose and 6 moles of oxygen. And obviously both the glucose and the oxygen are important to our survival as humans. So other things that plant cells can do for us, they can convert carbohydrate molecules to fat. A lot of the oils, obviously canola oil and corn oil, and then the generic vegetable oil that you might buy at the grocery store, these are all plant fats. And some can even convert these carbohydrate molecules into proteins. As animals, of course, we can't produce our own energy. We're reliant on plants as the ultimate source of our energy. And so either we eat the plants directly or we eat animals that have eaten the plants. So the plants are really in charge here in the food chain. And fortunately for us, they do a very nice job of catching that energy. And then we do a nice job of eating them or eating other animals that have eaten plants. So looking at the biological processes, we have metabolism, which is defined as the series of chemical reactions that keep a cell alive. If we want to be even more specific, we can break that down into 2 categories. There's catabolism, which is the process of breaking down molecules to produce energy, obviously an important thing for all of us animals, especially as humans, to be able to do. And then there's also anabolism, which is the process of synthesizing molecules. This is the building up part. And you've probably heard of anabolic steroid use or abuse among athletes. Baseball in particular has an unfortunate history with anabolic steroids. So anabolism is the building up, the making of these complex molecules. And then catabolism is the breaking down of these molecules to make energy. Both important parts of the metabolism processes, But again, those are the distinctions between the two parts. Alright, so looking specifically at the things that we can break down for energy, carbohydrates are one of the main types of macromolecules that we'll study. And carbohydrates themselves are polyhydroxyaldehydes or ketones. They're either aldoses or ketoses, as we'll classify them. And they can be hydrolyzed to form such compounds if they're not directly aldehydes or ketones. If they're in ring structures, for instance, we can hydrolyze them to make these carbohydrates. Monosaccharides are carbohydrates that cannot be hydrolyzed into anything simpler. So this would be like your glucose or your fructose, your simple monosaccharides. Most of the polysaccharides that we'll encounter can be broken down into these monosaccharides by chemistry. So as I mentioned, a couple examples of these monosaccharides are aldoses and ketoses are generic classes. So an aldose, as you look at the left-hand side, glucose and galactose are examples of aldoses. And if you look at the linear structure of these molecules, they end in an aldehyde. So they have differences in the specific values. The first carbon there that we see at the top is what classifies them as an aldose, because it's an aldehyde, right? It's a carbon bonded to a hydrogen with a carbonyl, a C double bond O, and then it's the rest of the carbon chain. So that's an aldehyde. And then if we go down to the fourth carbon from the top, we see it's the orientation of the OH group in the hydrogen that differs between galactose and glucose. But they're both aldoses because they both have that aldehyde functional group in their straight chain structure. If we contrast that with fructose on the far right, fructose, as you see, that carbonyl carbon is in between two other carbon groups. So there's a carbon group above and a carbon group below, so it's not an aldehyde, which requires the carbon to have a bond directly to a hydrogen. This is a ketone, where the carbonyl has the carbonyl carbon bonded to carbons on either side, and therefore fructose is classified as a ketose rather than an aldose. It's because of that ketone functional group. So most of the time, unfortunately, as we'll see in the next slide, the aldoses or ketoses are not in their straight chain structure. They're in a ring form, and that makes things even harder to look at. But hopefully with these straight chain structures, you can easily identify the key difference between aldoses and ketoses, and that's the presence of a ketone functional group in a ketose versus an aldehyde functional group in an aldose. Okay, so as I just mentioned, these monosaccharides are usually in their cyclic forms, their ring structures. They spend very little time, less than 5% of the likelihood of finding them as straight chains. 95% or so of the time they're found in these ring-shaped structures. And you can see there's still the difference between the glucose and galactose, which are still both, of course, aldoses, and the fructose, which is a ketose. If you look at that ring structure, the fructose has a 5-membered ring, the galactose and the glucose have 6-membered rings, and if we look at that OH-H difference, it's on the leftmost carbon of the ring. We see above the plane of the ring is the hydrogen, and below the plane of the ring is the OH group on glucose, and it's the opposite orientation in the galactose. So that's they're called anomers because they differ in only how they occur relative in terms of position above or below the ring. And then fructose, obviously a 5-membered ring, and that's more what we tend to see for ketoses. So now that we've classified the monosaccharides in either the aldose or ketose form, at least for the straight chains where we can see that clearly, let's talk about disaccharides. We'll complicate things a little bit by having now, instead of just a single monosaccharide, we have 2 monosaccharides that have been hydrolyzed, or actually dehydration synthesis is how we make the disaccharide. Then the disaccharide itself can be hydrolyzed back into the 2 monosaccharide units. So you can see with sucrose, which is our table sugar, sucrose is a glucose and a fructose molecule that those 2 molecules have combined to form a single sucrose disaccharide. It gives that classic sweetness that we're used to from table sugar. Lactose on the right is another example, and lactose, obviously, two different sugars there, galactose and glucose. Okay, so we see the oxygen there that bridges them from the dehydration synthesis of the disaccharide. So if we continue to synthesize larger and larger, more complex carbohydrates, so we go from the monosaccharide to the disaccharide to what we would call oligosaccharides, which are just smaller groups, all the way up to polysaccharides, where we have very large molecules that have lots and lots of monosaccharide units. So examples of these would be starch, cellulose, and glycogen. Starch tends to be the the starch is the plant storage form of glucose. Glycogen is much more highly branched, and that tends to be the animal storage form. And of course, cellulose isn't really a storage form of monosaccharide units for energy, but rather for structure. As we looked at the plant cell wall, that would be a cellulose structure. So depending on how we link those monosaccharides, we can get a storage form that's more linear for starches and more branched for glycogens, or we can get something that's structural, that's not meant to be used as a fuel, but rather for structure if we link the units in the way we do for cellulose. So polysaccharides, large complex molecules that can have very different applications depending on the particular linkages. And here we see the example of a segment of a starch molecule versus a segment of a cellulose molecule. And it's really the linkages that differ. So alpha linkage in starch, and then a beta linkage in cellulose. So as humans, we can break those alpha linkages quite well. So we can take starchy foods, for instance, and break them down into the glucose monomers, and that works really well for humans for stable blood sugars, because it takes us a while to break down the starches and to release that glucose slowly into the blood. If we look at the cellulose molecule, humans can't break that beta linkage, so that even though that's the same monomer, it's the same glucose monomer, we can't free it as humans. And you could be, eating cellulose-rich foods, but not be able to get really any energy from it, because that cellulose is more roughage. It's the cell walls of plants and things that we can't digest. It's important forms of fiber, but it's not anything that we can use for energy, because humans lack the enzymes to break that beta linkage. So if humans can't use cellulose for energy and plants can't use cellulose for energy, well, then why do we have cellulose even in existence? Well, of course, cellulose is important because it makes up the structural units of plants. Those cellulose chains are composed of parallel bundles called fibrils, and these allow the plants to have their rigid structure and to be able to support themselves. If you think of something like a stalk of celery, there's a lot of cellulose there that gives that rigidity and structure to the celery stalk. So it's not important for the energy that's possible. Obviously, if we burned it, we could release the energy that's stored in those chemical bonds. But when I say burned it, this would be combusting it in a fire. We can't burn it in the sense of getting the fuel out of it like we can for starch or glycogen as humans. But that cellulose is important nonetheless, because it's important structural features for plants, and then it's important for humans as dietary fiber that helps to move things along in our digestive system. When we use the term starch in plants, there's actually two polymers that we're talking about. There's amylose and amylopectin. In amylose, the glucose molecules are connected in long parallel chains. In amylopectin, the chains are highly branched, and therefore they give rise to different structural forms. In either form, humans and other animals can break down the starch to release the glucose monomers, something we can't do with the cellulose that we just talked about. But of course, either form is useful for us, although as we'll see, the amylopectin is a little more like the glycogen that we use as humans. And here it is, a glycogen, which is sometimes known as animal starch. It's similar to amylopectin because the glucose polymers are branched, and this is really something that we have stored in our muscle cells, for instance, to be able to get that burst of energy, that long term burst that we might need for something like a marathon running, where we need to have that controlled amount of glucose, but we're not going to be able to be stopping and eating as we're running the marathon. We need that glycogen to be in reserves so that that slow release of glucose can continue while we're doing something very energetic like running a marathon. So for humans, the way we can store glucose is as those highly branched glycogen molecules. And here we have a nice image of those three different forms of glucose as a polysaccharide. So amylose, where the glucose molecules are really pretty much in a straight chain. Amylopectin, where you have some branching to the chains. Both of those are starches that you'd find in plants. And then finally the glycogen, the so-called animal starch, is much more highly branched, even more highly branched than the amylopectin. And again, these are just three different polysaccharides which are made up of these glucose monomer units. Okay, so the carbohydrates are important for the energy use in cells, and as animals and humans in particular, our brains can only use glucose as a fuel. So that glucose obviously is important, and a lot of the other monosaccharides can be rearranged to form glucose or can be burned in other pathways. But if we really need to be able to store large amounts of energy, the glycogen, again, is short-term storage for cells to be able to access glucose. If we want long-term storage, we're looking at the fats and lipids. So lipids are biomolecules that are insoluble in water, but soluble in nonpolar organic solvents. Fats are specifically esters of these long-chain fatty acids and glycerol. And so we'll call them triglycerides or triacylglycerols because glycerol itself has those three OH groups, if you remember from our discussion of organic chemistry. Each of those OH groups can undergo condensation, polymerization with a fatty acid to cause the formation of a triglyceride. So that's what we'll be looking at here for these high energy storage molecules known as fats. Of course, fats get a bad rap. Everybody wants to cut fat out of their diet. And again, having too much fat in the diet is a problem, but we do need some fat. We need to be able to have these ways of storing energy for later use in these high storage fats instead of just relying wholly on glycogen, for instance. Here we see table 16.1, some fatty acids found in natural fats. The shorter ones we tend to associate with animal fats, so 4-carbon or 6-carbon, butyric acid or caproic acids. These are found in butters, and they're actually quite unpleasant odors as the free acids. So if you've ever smelled vomit, that's a mix of butyric acid, mostly butyric acid, maybe a little caproic acid, and it's a very unpleasant odor. A good way to neutralize that odor quite chemically correctly to say neutralize, because you can take that acid and take a strong base like lye or even ammonia and remove the OH group, and you'll form the sodium salt in the case of lye, and that would give you a very effective way of removing that odor, that unpleasant vomit-like odor. And as we see, as we move up, we've got some other longer chains ones that we may find in other sources. Oleic acid in particular, that 18-carbon monounsaturated acid, and again, that's the first one. If you look, you see the double bond. That's what we mean by unsaturated. If you go back to our discussion of organic chemistry, unsaturated means carbon-carbon double bonds. Saturated means all carbon single bonds. And then we have linoleic acid, which is 2 unsaturations, and then linolenic acid has now 3 unsaturations. This is what we get into where we talk about polyunsaturated. Arachidonic acid has 4 unsaturations, for instance. So monounsaturated fats would be things like oleic acid, which is typically found in olive oil as its principal source. But then you've got other things like fish oils and liver oils that have polyunsaturated fats. Here we see palmitic acid, which is a very long chain fatty acid. We see the polar head group, that carboxylic acid head group. Of course, that's why we call it an acid. The C double bond O, single bond OH. That's a polar head group, right? There's hydrogen bonding that can happen there that makes it nice and polar. And then you've got that long chain of the fatty acid that's non-polar, just carbons and hydrogens there. And so we would see that if we were to arrange these things, the non-polar tails would all be attracted to each other, and the polar head groups would be facing out toward the water that would be present in any cell. So we tend to have a polar portion, a polar head group as it's called, and then a non-polar tail for these fatty acids. Going back to that idea of fats as triglycerides, that's because, as we see up above, we have glycerol, that 3-carbon triol. It's a trialcohol, 3 OH groups off of the 3 carbons, 1, 2, 3 triol. And then each of those OH groups can form an ester bond with an OH group from a carboxylic acid to form the triglyceride. And as a result, of course, you get 3 waters as the coproducts. So we see here an example in B, a sort of space-filling model of a triglyceride where all of those fatty acid ends are together to the right of the molecule and all of the polar head groups are together on the left of that triglyceride. So thinking back to our organic chemistry, saturated fatty acids have no carbon-carbon double bonds. They're all carbon single bonds, and we saw examples of those were things like butyric acid. For monounsaturated fatty acids, you have at least, or I should say only one carbon-carbon double bond, and that was like oleic acid. And then finally, polyunsaturated fatty acids have 2 or more carbon-to-carbon double bonds, and these would be things like arachidonic acid that had the multiple C double bond O groups. So if you think about solid fats, things like butters and other animal sources of fats, lard for instance, they tend to have a high proportion of saturated fatty acids. So butter, we said, has a lot of butyric acid, which was a short chain, just a 4-carbon saturated fatty acid. Liquid oils, on the other hand, tend to have unsaturated fatty acids. So olive oil, which is mostly the oleic acid residues, those have that 1 unsaturation between carbons 9 and 10 on that 18-carbon chain. As a result, you tend to see, as I said, mostly animal fats are solids, although coconut oil, we call it an oil even though it's mostly a solid, you tend to have a low chance of having them go rancid because rancidity often affects the double bonds. So oils can go rancid much more easily because of the unsaturations. That's where the reactivity is. For solid fats, things like butter, of course, we don't want to have too much butter in our diet because these saturated fats have a tendency to be linked to heart disease, but you do have a lot of stability in those saturated fats, so they don't tend to go rancid as long as you're refrigerating them and things like that. Whereas oils, polyunsaturated fats in oils, for instance, can go rancid pretty quickly, and they have to be kept under refrigeration whenever they're not really actively being used because of the reactivity of that unsaturated carbon-carbon double bond. And as chemists, one of the ways we keep track of the reactivity, the number of unsaturations, is by iodine number. Iodine number is a measure of the degree of unsaturation of a fat or an oil, and technically speaking, iodine number is the number of grams of iodine that are consumed by 100 grams of a fat or an oil. So if iodine can add across a double bond, as we see in the reaction at the bottom of the slide, the more double bonds you have, the more iodine will be used up by reaction with that fat or oil. So we see here in table 16.2 some typical iodine numbers, and as you see, the low iodine numbers, coconut oil, as I said, is sort of a misnomer. It's not really coconut oil. It's a solid fat like butter, and it has a very low iodine number. There's very few unsaturations in coconut oil. Butter, beef tallow, lard, as we see those traditional animal fats, they have very low iodine numbers. If we compare things like safflower oil and sunflower oil toward the bottom, those are polyunsaturated fatty acids that go into making those oils, and you have very high iodine numbers as a result. So it's good to have some of the polyunsaturated, good to have some of the monounsaturated, and it's even good to have some of the saturated fats in a healthy, balanced diet. But if we're looking at the oils, the true oils, the liquid oils tend to have large unsaturation numbers because they have, as you see, large iodine numbers that go hand in hand, and the animal fats tend to be much lower iodine numbers because they tend to be mostly saturated fats, relatively few unsaturated fatty acid residues. Okay, so far we've talked about carbohydrates and lipids, and as we saw, both those classes of macromolecules tend to be used for energy storage and production, although as we saw with cellulose, that was a carbohydrate that actually had more of a structural function. Now that we're looking into proteins, proteins can also be metabolized, and if you look at your nutritional labels, you'll see carbohydrates, which they'll commonly call sugars, and then lipids or fats as they might put them on the label, and finally proteins on the label. They will have calories associated, and they can be used for energy storage, but proteins are much more like the structural carbohydrate there, the cellulose, because the proteins tend to have non-energy storage uses. Proteins are largely used for a number of different applications, and they are a vital component of all living cells. Proteins are critical to the flexibility and diversity of life that we see because of the flexibility of the way proteins can form from the amino acid side chains that exist. All right, so explaining that protein diversity, proteins are polymers of amino acids, and as I said, amino acids have a variable side chain, but they also have the amino group and the carboxylate group, the acid group, attached to that same carbon, which we call the alpha carbon. So there's a carbon that has an amino group and a carboxylic acid group attached to it. It also has a hydrogen, and then it has some variable group that gives us the specific identity of the amino acid. That R group, as it's called, can be as simple as a hydrogen for glycine, or it can be fairly complex for other amino acids. And depending on the identity of the R group, you're going to have more of a nonpolar or more of a polar side chain, and that's going to affect how proteins fold and what function the protein can perform. So very important stuff based on that simple little structural feature of the R group. Here we see two amino acids that have been joined together through that amide or peptide linkage, as it's sometimes called. The biologists tend to be the ones using that peptide language. The chemists prefer to use the amide group because we saw that back in organic chemistry, that carbonyl bonded directly to a nitrogen was known as an amide. And obviously there's still a free amino group on the one amino acid and a free carboxylic acid group on the other, because the way that they joined, the carboxylic acid end of one and the amino end of the other came together to form that amide linkage, and that's our two amino acids that have undergone this dehydration synthesis. They kicked out of water and they made that peptide linkage. So in addition to being able to link together to form these polypeptides, these multiple peptide linkage, or really amide linkages, amino acids can also exist as dipolar ions or inner ions, depending on that physiological pH. We call this Zwitterion. Depending on how good your German is, you may have more of a W or a V sound there, but as we see, that amino terminus, the 3 amino group on the one end, has been protonated to form an NH3 plus, and then the carboxylate group on the other, the carboxylic acid, has been deprotonated to form a carboxylate ion. So instead of having the COOH and the NH2, that COOH has lost the H plus to become the COO minus, and the NH2 has gained the H plus to become an NH3 terminus. So we've got, instead of a neutral molecule, we have di-ion, a Zwitterion, so to speak, at least for this glycyl phenylalanine dipeptide. And it's a dipeptide because it's a glycine and a phenylalanine group, 2 distinct amino acids that have been joined by that peptide linkage. Okay, so we mentioned that we're dependent on the plants for the oxygen and for the carbohydrates. Ultimately, they're the source, whether we eat the plants directly or eat animals that have eaten those plants. They're the energy producers, and they're also the oxygen producers on Earth. Plants are also important because they can synthesize protein from carbon dioxide, water, and minerals with things such as nitrates or sulfates. As animals, we have to consume proteins as part of our diet. Humans can synthesize some amino acids, but there's the so-called essential amino acids that we have to get from our diet, and they're called essential because we cannot make them. We have to get them from food sources. Animal sources tend to have a lot of the essential amino acids. They are what we call complete proteins, but if you're a vegetarian, you don't have to eat animal sources in order to get those amino acids, but you do have to be careful to blend your sources because you run the risk of not getting all of the essential amino acids if you don't have a balanced diet. So things like legumes, beans, for instance, tend to be a nice protein, and you can combine that with protein sources, other plant protein sources, to make sure that you get a complete group of the essential amino acids. Whereas if you eat animals or animal products like eggs or milk, you're more likely to be able to get those essential amino acids without having to do too much extra work figuring out how to blend your diet to optimize those. Okay, as we've already mentioned, amino acids can be linked by one another to form these polypeptides, these proteins, by making an amide linkage between the amino terminus of one amino acid and the carboxylic acid terminus of another. Again, in chemistry, we recognize that it's an amide linkage, and if you remember your organic chemistry, we should have no trouble seeing that, but because of its early name of peptide, we'll sometimes call this a peptide bond. And again, that tends to be more clung to in the biological circles than in the biochemical circles. It's really an amide linkage, which is possible in living systems through the peptide bond, or in non-living systems we have lots of amides possible in certain synthetic fibers, for instance. They're amides. So as chemists, we can call it an amide linkage, but just realize that in biological circles, you're much more likely to still see it referred to as the peptide bond. And we see here a tripeptide, where we have 3 amino acids that have come together in just such a way that we had a serine and then an alanine and a cysteine linkage that we call now cysteine, which is a tripeptide. And the naming does tell you from amino terminus to carboxylate terminus the order of the 3 amino acids that went into this tripeptide. Okay, so getting the language down, we've already seen a few examples of dipeptides, which are formed when 2 amino acids are joined. And actually, the sweetener that's used in a lot of diet beverages, for instance, or at least historically has been used, is an example of a dipeptide. So it happens to be the phenylalanine and aspartic acid. That when you combine those two in dipeptide fashion, you get something that is very, very sweet, much sweeter than sugar, the sucrose, table sugar. And that was an accidental discovery, and it's been used for a long time, although you have to be very careful if you're a phenylketonuric. If you can't process phenylalanine, there's a warning on the products that have this, because of course phenylalanine and aspartic acid are the two amino acids that go into it, and obviously if we metabolize it, you can have a buildup of phenylalanine that can be very damaging for someone who suffers from phenylketonuria. That's an example of a dipeptide that happens to be very sweet and has been used as an artificial sweetener. Tripeptides would have 3 amino acid units. Polypeptides would have 10 or more amino acid units, up to about the lengthy proteins, which can have 10,000 or more amino acids. So proteins are even larger than polypeptides and can have many amino acid units linked together. As I mentioned when I was naming some of those interesting sounding dipeptides or tripeptides, the sequence of the amino acids is very, very important. So the sequence is always named from the free amino group, the N-terminus, or the N-terminal as our book says, to the free carboxyl group, the C-terminus, or C-terminal as our book says. And so that way we can have our order correct, and we can be talking about the same thing. Even in that aspartame example that I gave, it makes a big difference whether the aspartic acid residue has the free nitrogen terminus and the phenylalanine has the free carboxyl terminus, or the other way around. If we switch the order, then you don't get the sweetness, you don't get the same substance. So order is critical in proteins, because the way that our biochemistry works is dependent on which goes first, which has the free amino terminus, which has the free carboxyl terminus. So that order of the amino acids in the polypeptide or in the protein, again, important for structure and function. So the primary structure of a protein is really just that sequence of amino acids starting at the nitrogen, the amino terminus, and working our way to the carboxyl terminus. So for the primary structure of angiotensin II, for instance, we have an aspartic acid residue, an arginine residue, a valine residue, a tyrosine residue, an isoleucine residue, a histine residue, a proline residue, and then at the carboxyl terminus we have a phenylalanine residue. If we were to put it in just the opposite order, where the phenylalanine were to have the free amino terminus instead of the free carboxy terminus, and the aspartic acid residue would have the free carboxy terminus instead of free amino terminus, we'd have something that no longer functions as angiotensin II, because the order is switched. In terms of the primary structure, we have the wrong sequence. Now, the reason that primary structure sequence is so important is because that's what determines the later structures. The secondary structure, for instance, depends on that primary structure sequence to allow for now the folding and coiling that can happen as a result of hydrogen bonding and other interactions in the backbone of that amide linkage. So examples of some secondary structures include alpha helices and beta pleated sheets, and these things happen because the primary structure allows for it, because you can bend back on yourself in such a way to maximize those intermolecular interactions. So we see in this particular picture a beta pleated sheet moiety. Moiety is just a fancy way to say structural feature. And so if we have the primary structure that we see here, and then it goes and bends around itself and then links back, now we've set ourselves up for optimal hydrogen bonding between the NH groups of the top sheet and the carbonyl groups of the bottom sheet. So you have the partial negative at the oxygen end of the carbonyl, you have the partial positive at the hydrogen end of the NH, and those allow for some really nice, strong intermolecular hydrogen bonding that gives rise to this beta-pleated sheet array. Another very popular secondary structure of proteins is the alpha helix, and that also results from intramolecular hydrogen bonding, but now it's because as you twist, you're able to spiral around again to hydrogen bond. And this was Linus Pauling, who was a brilliant scientist, a chemist who discovered most of the structural features of proteins. And he reportedly discovered the alpha helix while he was sick one day. He was stuck in bed, and he was quite ill and couldn't report to his lab, so he asked for a pencil and paper, and he was just from memory sketching the backbone bonding on the sheet, and he was able to discover, because of his brilliant mind, that if he were to twist in just such a way, he could maximize hydrogen bonding, and that's how he reportedly came up with the alpha helix. Now, whether that's a true story or a bit romanticized, like Newton with the apple falling on his head, we may never know, but he certainly is the individual credited with all of this, and that's the story he tells as to how he discovered the alpha helix array. Alright, so our primary structure, again, just the straight chain amino acid sequence in our polypeptide. The secondary structure, a result of that straight chain starting to bend and interact back with itself, usually through things like hydrogen bonding, as we saw for beta-pleated sheets and for the alpha helix. Now, if we look at tertiary structure, now we've got our coiled coils. So the secondary structure started us bending back on ourselves, and now tertiary structure will allow for even further bending and structural array based on that three-dimensional shape, being able to maximize those spatial relationships for different side chains at different parts of the protein. And one of the key examples that we see in our picture here is globular proteins. So the protein chain in a globular protein. Those of you who are as old as I am probably remember the old telephone. In our day, at least, we had telephones that had cords. We didn't have cordless phones until a little bit after I was born. And so you had the phone cord, and the phone cord itself is sort of like an alpha helix, if you know what a phone cord looks like. And because of use, the phone cord would not only have that original coil, but it would coil on itself. And that's what these tertiary structures are. It's the coiled secondary structures that start to interact with each other again, as we see here in this heme group of this globular protein. And now finally, we've worked our way up to quaternary structure, this highest level of structure of proteins, and that involves the interaction of more than one peptide chain. So we have a tertiary structure, like we saw in the previous slide, of one protein chain, and then you have another subunit, or in this case, three other subunits that come together in such a way that they are able to interact in 3D space to form this overall protein. So each individual protein chain has its primary, secondary, and tertiary structure, and then these four different subunits come together to give rise to the quaternary structure, the final overall structure of the protein that allows it to function in the way that it does. And this is really intricate and elegant chemistry. Biochemistry, indeed, I would say, is the most elegant of all the chemistries because of all the work that goes in to making these really four different active sites. So thousands and thousands and thousands of side chains to get four active sites in a protein. It's just really incredible work that biology has done for us in the way that we've made these things like the heme groups in hemoglobin protein. Alright, so trying to summarize something as complicated as protein structure, let's try to do our best. And I'm no biochemist, I'm an inorganic materials chemist, but I do have a fascination with biochemistry, and I hope I've instilled a little of that with you in this chapter, because it is really interesting and elegant and beautiful chemistry. And it's based mainly on these 4 ways that we can link these protein chains. The first and the strongest interaction that's not a true bond is the hydrogen bond. We call it a bond, but it's not truly a bond. It's an intermolecular interaction, and it occurs when secondary structures can form hydrogen bonds between typically amide hydrogen NH groups and carboxyl oxygen groups. Tertiary structures will also involve hydrogen bonding between the side chains of the amino acids. So a number of those amino acids are side chains that can participate in hydrogen bondings because of OH groups, for instance, or NH2 groups. Ionic bonds, these are true chemical bonds. They're also known as salt bridges. They can occur between oppositely charged side chains if the side chains can develop a full positive or negative charge. And again, this can be as well because of carboxylic acids that get deprotonated or amine groups that get protonated as side chains. That's how you typically will see your ionic bonds or your salt bridges. Disulfide linkages, these are specific to cysteine side chains. So when the 2 cysteine side chain residues can be oxidized, you can make an SS single bond. A disulfide linkage can form, and this is often, if you think about curly hair, people will go and get their hair permed to try to force a curl to otherwise straight hair. Oftentimes it'll have that sort of sulfurous odor. That's because cysteine residues are involved. You're trying to force these disulfide linkages. And finally, dispersion forces, which are individually quite weak, but when you get lots and lots of them, you can have something fairly strong, and you can make these nonpolar side chains form little pockets of nonpolar interaction, which arise from these attractive forces between the 2 nonpolar side chains, or more than 2 depending on the complexity of the protein. That's dispersion forces, which, as I said, they are not as strong as hydrogen bonds on a per interaction basis, but if you have lots of them, then you can have a significant interaction. And we see here a picture, more a cartoon. And cartoon's not a bad term. I'm not trying to be negative here. I'm just trying to say that biochemistry gets so complicated, especially protein chemistry, that it's often simpler to have just the key structural features highlighted. So you see here we have just sort of a ribbon to be the backbone, and then we've highlighted each of these types. So at the far left, you see the hydrogen bonding that can happen between residues that have an OH group, for instance. They can hydrogen bond with the carbonyl of the amide linkage of the backbone of the protein. Then as we move to the right, we see an ionic bond, a salt bridge that can form between an amino acid residue that has a carboxylic acid group and an amino acid residue that has an amine group. They can form salt bridges across from each other as long as the protein twists in just the right way. Then we see that true chemical bond, that covalent bond. Again, an ionic bond is also a chemical bond, but the covalent bond that forms with the disulfide linkage that can hold those 2 side chains together. And finally, the dispersion forces that occur when you have nonpolar side chain interactions. Now, because biochemistry tends to happen in water, right? Cells are the fundamental unit of life, and cells are mostly water. We tend to operate under the principle of nonpolar in, polar out. So proteins tend to be arranged with all their nonpolar groups facing inside the protein so that they can interact with each other. And then the polar groups tend to be on the outside to interact with the water molecules that are inevitably surrounding everything in the cell. So you've got your nonpolar pockets, the hydrophobic pockets on the inside of the protein, and then the polar surface of the protein to interact positively with all that water. So proteins serve a number of important functions, some structural, some more chemical, and enzymes are one of the key biological catalysts, and enzymes are mostly protein based. So many of these protein-based enzymes are highly specific. They catalyze just one single reaction or a small related group of reactions, and that specificity is important for living things. We have to be able to catalyze a specific reaction because we don't want to have something so general that the enzyme works on anything and therefore works on everything. So we have to have specific reactions that can dictate what sort of biochemical processes can go forward. The substrate is what the reactant molecule that the enzyme catalyzes the reaction on, and typically the substrate fits very nicely into the active site of that proteinaceous enzyme. Some of these enzymes catalyze reactions that are helpful for building things in our bodies. Some break down substances that can be toxic to the body. So, but they all do very important functions, and they do them much faster than they would happen on their own. A lot of this chemistry would take a long time to happen on its own, but in the presence of a protein-based enzyme, they can happen very, very quickly at room temperature and conditions that are favorable to cells, rather than going high temperature, which human cells can't deal with high temperatures. The protein-based enzymes will let that chemistry happen in very favorable conditions for our cells. So, as I said, the substrates work well because they fit nicely into the active sites, and this is often called an induced fit model. The substrate molecule binds to the enzyme at the active site. They form this enzyme-substrate complex, and then the complex can catalyze the reaction of the substrate to form its products. So the enzyme plus the substrate combine in such a way as to make this enzyme-substrate complex, and then that enzyme-substrate complex can break back down to free the enzyme to work on another substrate, and that's what makes that enzyme so effective, is that it has what we call turnover. It can go again and again and again, and each time it takes that substrate, it makes that enzyme-substrate complex, and then it makes the desired product. It takes, for instance, a substrate like hydrogen peroxide that's very toxic to our cells, and it breaks it down into water and oxygen, things that are very helpful in our cells. So here we see a nice little cartoon of that process. The substrate approaches that active site of the enzyme. It doesn't look like it's going to quite fit perfectly, but as it approaches, the enzyme moves a little bit and allows the substrate to get in there. You make that enzyme substrate complex, and then at the end, that substrate is now changed. It's broken down into two products, and the enzyme is released again to work on another substrate. So that's what we mean by catalyst. It allows the reaction to happen, but it's not changed by the reaction. It changes in the course of the reaction to make that enzyme substrate complex, but at the end of the reaction, that enzyme is ready to go again and catalyze another breakdown of that substrate. So sometimes, unfortunately, enzymes get inhibited. So inhibition means that a molecule binds to the enzyme protein at some site other than the active site usually, and by binding in that allosteric site, that other site, it causes the active site to change its shape, and now it no longer accepts the desired substrate. So this sounds like a bad thing, and oftentimes it is a bad thing, but sometimes this is actually how drugs work. A lot of drugs are enzyme inhibitors, and if you've ever listened to those drug commercials that seem to be on TV all the time, I don't know if it's such a good idea for the drug companies to market directly to the consumers instead of maybe the physicians who might know a little more about it, but that's the world we live in, and oftentimes you'll hear what's even more concerning, I guess, is they'll say it's believed to act by, which tells you they're not even sure, but oftentimes they'll have a statement like, it's believed to act by inhibiting the COX-2 enzyme, for instance. That's a common enzyme inhibitor target. So these things can be undesirable, although oftentimes as drug targets, it's often desirable to inhibit certain enzymes that may be either overactive, or the products they make may be causing problems for certain patients' biochemistry. And so here we see a cartoon where you have the enzyme and the substrate, and then that allosteric site someplace outside, away from the active site of the enzyme. When the inhibitor binds that allosteric site, now the whole enzyme structure has changed, and the substrate no longer fits into the enzyme active site because of the presence of the inhibitor. So, as I said, this can either be an undesirable situation that can arise. Certain poisons, for instance, might act by inhibiting protein enzyme function, or it can be a drug target to alleviate some problem, some overactive um, process that's causing trouble for the patient. But it's a very nice cartoon of how we believe that inhibition to work by binding at an allosteric site. Sometimes enzymes require another molecule in order to function correctly. Cofactors are these substances that can be either inorganic ions, like zinc ions, zinc finger domains, for instance, you might hear oftentimes, or magnesium ions for things like chlorophyll, or even just small organic molecules that need to be present for the enzyme to function. Oftentimes even vitamins may be cofactors for certain enzyme function. Coenzyme is another a cofactor that's a non-protein organic molecule. So coenzyme might be something like vitamin A or vitamin E, for instance, depending on the particular protein. And finally, apoenzyme is a term we use when we're talking about the pure protein part of an enzyme without the cofactors or coenzymes. That's the term that's used. So some things you might have seen or you might run into, and hopefully this clears up any confusion. Our understanding of enzymes has allowed for a lot of pretty rapid, easy-to-use tests. For instance, diabetic test strips use 2 enzymes to measure blood sugar. As we see, one enzyme will cause the oxidation of glucose and produce hydrogen peroxide as a byproduct, and then that other enzyme breaks down the hydrogen peroxide and oxidizes a dye to give a color change, and based on the color that you get on your strip, you have an indication as to what the blood sugar level is, whether it's in the normal range or whether it's too high or too low. And that's important for a diabetic to be able to measure quickly and respond accordingly. Either get some more sugar in if the blood sugar is too low, or just wait before eating if the blood sugar is too high, or do nothing if the blood sugar is right where it should be. You can also use enzymes to monitor liver damage or heart damage and to diagnose diseases as a result. Finally, you can use them to break up clots after a heart attack or increase clotting to treat hemophilia, which is a blood clotting disorder. So enzymes are very important and have been used for quite a while now in medicine and healthcare applications, and certainly we'll see more of that as we come to understand more of the biochemistry of enzymes. Enzymes are also an important part of the chemical industry, especially food industry applications. Production of baby foods, beer, the production of beer, these microbreweries will often use enzymes to help move the processes along. Sweeteners for soft drinks, animal feeds, even things like blue gene dyeing and production can involve the use of enzymes. So enzymes have become a very important part of not only traditional biochemistry and biomedical applications, but other applications as well. Because they're largely protein-based, enzymes have a lot of desirability in terms of making specialty chemicals, new drugs, even to break down complex pollutants in the environment. The enzymes seem to be a very green route to doing things that maybe traditional chemistry has resulted in hazardous waste production, or just byproducts that are not environmentally friendly. Enzymes can often provide a pathway that's a much more environmentally friendly solution. Enzymes are also useful in everyday life. They're used in applications like stain removers and meat tenderizers. If you happen to be an individual who's lactose intolerant because your body doesn't produce the lactase that other people who can tolerate lactose have, you can take enzymes to reduce that discomfort. You can take that lactase as an outside enzyme and therefore digest the dairy foods and be able to have ice cream and milk and things that maybe without that enzyme that you take as a pill, you wouldn't be able to enjoy, or at least you wouldn't be able to enjoy without a fair deal of discomfort. as you see in our slide, worldwide production of enzymes is worth more than $1 billion per year. So it's big business, and it's helping improve the lives of many people who take these enzymes. Okay, so far we've discussed the carbohydrates, lipids, proteins and protein-based enzymes, and now the last of the major four macromolecules are the nucleic acids. And nucleic acids are not typically used for energy production or any of the things that we saw the other macromolecules being used for. These serve as the information and control centers of the cell. And we typically have the two major forms. There's deoxyribonucleic acid, DNA, and ribonucleic acid, RNA. And they're similar in that both consist of long chains called nucleotides. Each nucleotide is composed of a sugar unit, a phosphate unit, and a heterocyclic amine base, which we'll come to classify as either a purine or a pyrimidine in the coming slides. As you can see in this slide, we've got nitrogen bases and sugar and phosphate components that we may see in DNA only for thymine, for instance, and RNA only for uracil in terms of the nitrogenous bases. For DNA, obviously the sugar and phosphate component is 2-deoxyribose, and then in RNA it's just plain old ribose. The phosphate is present in both DNA and RNA, and for nitrogen bases you have adenine, guanine, and cytosine being present in both RNA and DNA, with again thymine and uracil distinguishing RNA and DNA, thymine for DNA, uracil for RNA, and then obviously the sugar, 2-deoxyribose for DNA and ribose for RNA. So when looking at nucleic acids, nucleotides are important structural subunits, and they're composed of a sugar, a phosphate, and an amine base. And so we see here adenosine monophosphate is our nucleotide, and the phosphate is linked by what's known as a phosphate ester linkage. So a traditional ester is a COC bond. Here a phosphate ester is a COP bond, with that phosphate group being the rest of the phosphorus moiety. And then finally we have attached to that sugar is the amine base. DNA, of course, is made up of a double helix, the classic text, The Double Helix by Watson and Crick, where they elucidate DNA's structure. That's another story where we probably should have been talking about Linus Pauling. Linus Pauling won the Nobel Prize for Chemistry, one of the few people who won it solo, just to give you an idea of how important his contributions were for all of his work with proteins, protein primary, secondary, tertiary, and quaternary structure. And Pauling was very close to decoding DNA, but he didn't get to go to England to see the X-ray images that Watson and Crick got to see. And the main reason he didn't get to go is because he was an outspoken critic of war. He was actually also a Nobel Prize winner for peace, one of the few people who won in multiple categories. Chemistry, obviously, for his work on proteins, and then peace for his anti-war activities. But it was those anti-war activities that caused him to be denied a travel visa to go to the conference in England that Watson and Crick were able to attend, and it was at that conference that they got to see some X-ray images that allowed them to discover the double helix structure before Pauling did. So again, a bit of an upset here. Otherwise, all the major biochemical discoveries would have been made by chemists instead of molecular biologists like Watson and Crick, but that's history, and I guess we can't change it. But the take-home story is DNA is a double helix, and its double helix structure allows for it to be so conveniently bound there in the nucleus in these really tight chromosome structures. So the double helix of DNA is held together by base pairing, and we call them bases because they're nitrogen bases. They are basic in terms of chemistry as opposed to acidic, and it's the complementarity that allows these bases to join and hold together that double helix. if we look, thymine and adenine, that AT grouping allows for 2 hydrogen bonds. There's the hydrogen bond there from the amino group there at adenine, and also the nitrogen that's part of that ring can hydrogen bond with the hydrogen from a nitrogen that's part of the thymine ring. If you look at cytosine and guanine, now you have 3 hydrogen bonding groups possible. So the CG residues actually have a stronger interaction than the AT residues. If we look at thymine and cytosine, we see that they are both known as pyrimidines based on their ring structure, their single ring structure. Adenine and guanine are both purines because they're fused rings. There's a 5-ring fused to a 6-ring for those, and that's based on the structure of purine. So you have a purine and a pyrimidine interacting across the double helix, and the adenine-thymine has 2 hydrogen bonds. Cytosine-guanine has 3 hydrogen bonds, and that's important if you're trying to disrupt the DNA. That's going to require a little more energy to disrupt those CG interactions because of the 3 hydrogen bonds as opposed to just the 2 hydrogen bonds in an AT interaction. Looking at the structure here of RNA, RNA consists of single strands of nucleic acid. So different from that DNA double helix, we now have single strands. And RNA is much more reactive as a result. DNA, we can collect DNA from samples of living organisms from thousands of years ago because DNA is a long-term storage molecule. It allows for things to be conserved. We don't want changes to DNA, right? Changes to DNA can result in things like cancers and undesirable effects. DNA is a stable long-term storage type molecule. RNA, on the other hand, is much more reactive, much more flexible, being these single strands of nucleic acid. So I mentioned chromosomes a couple of slides back, and chromosomes themselves, of course, are these thread-like strands of DNA that are tightly coiled into mostly X-shaped bodies, right? There's the Y chromosome that has more of a Y shape if you're a male, but the human body contains 46 chromosomes, 23 of which came from the egg of your mother, and 23 of which came from the sperm of your father, and then those, for the most part, they're duplicates of each other, aside from males have just one Y chromosome from their father and an X chromosome from their mother, whereas females have an X chromosome from both the mother and the father. A gene is that section of a DNA molecule that actually controls for the synthesis of a protein. And so that's all DNA is for, right? It's just a matter of synthesizing proteins. It's a lot more involved than I made that sound, but that's what it is. And if there's damage to a gene, then the protein doesn't get made or doesn't get made correctly, and that's where we run into problems. And that's where ultimately things like cancers can result from an abnormal gene. Replication is the copying of DNA during cell division, and we have lots and lots of controls trying to make that as faithful a process as possible, high fidelity replication, so that we don't get DNA damages or DNA copying during that replication process. Obviously, before the cell divides, each of these new cells that's going to form has to have a complete set of the DNA in order to do their protein synthesis. And if we look here, that DNA double helix unravels a bit to allow for each of the strands to make a new copy. And so that double helix of the original DNA now allows for 2 double helices to be formed, an old and a new. And there are DNA polymerases that bring in those nucleotides to make the new replicates. And there's some other things that we don't show in this simple cartoon, things like chaperonins, which are proteins that make sure that everything goes right, just like a chaperone at a dance makes sure things don't get out of hand. That's what we have our chaperonins for in DNA replication, and lots of neat things happening. And we've been able to figure out a lot of this elegant chemistry and use it for things that we'll talk about toward the end of this chapter. As I've said already, that DNA is the long-term storage of the genetic information. So it's important that that molecule be conserved and it's kept in the nucleus. However, DNA is also there to guide protein synthesis, and the way that that happens is by transferring information from the DNA to RNA by a process that's called transcription. So during transcription, the messenger RNA is synthesized, or we'll call it mRNA from here on out, but that's just a shorthand notation for messenger RNA. So looking at table 16.4, we see in DNA we have adenine, thymine, cytosine, and guanine. In the complementary RNA, we have uracil replacing the thymine. So uracil is another example of a pyrimidine, but it's different than the thymine that we would see in DNA. Uracil is the RNA complementary base. So instead of AT, like we would see in DNA, we see AU in RNA. Table 16.5 gives us our genetic code, so what that messenger RNA is coding for. For instance, if you have 3 uracil residues, that's coding for a phenylalanine. Likewise, a uracil cytosine is also phenylalanine. So there's some redundancy here. There's the various amino acids, and there's a couple of different codes that might code for a given amino acid. And so we see that here in table 16.5. So if you were paying attention there to table 16.5, you notice that it was a 3-letter code. So the genetic code is carried on a sequence of 3 bases known as a codon. And so the codon specifies a given protein by base pairing with the anticodon with a specific transfer RNA. So we have the messenger RNA, now we have transfer RNA through this process of transcription. So that's how we take these genetic codes and translate them into actual amino acid building blocks. And here we see another cartoon where we have the DNA, which has the messenger RNA that interacts with it for transcription, and now we have the transfer RNA come in and actually put the sequence of amino acids together based on these codons and anticodons, and we get translation. We get the making of proteins at the ribosomes by having that DNA stored instructions that's accessed by the messenger RNA and now actually translated by the transfer RNA into a protein chain. And that's the whole process where DNA codes for protein synthesis by allowing the messenger RNA and the transfer RNA to do their job based on that DNA code. Very elegant chemistry, very good system. It's worked well for biology for many, many millennia. And now humans have started to tinker with it as well, because we just can't seem to help ourselves. When the chemistry's this good, we want to get involved. Okay, so one of the ways that humans have gotten involved with the genetic code is by genetic testing. So one DNA sequencing technique that we use is a polymerase chain reaction known as PCR. So in this technique, the DNA is cleaved by enzymes. Bacterial enzymes called DNA polymerases are used to multiply the number of DNA fragments. So we're just making more and more copies. If we had a very small amount to begin with, let's say in a forensic type setting where you have a limited amount of DNA at a crime scene, you can use PCR to multiply that. And now these fragments can be separated from longest to shortest. You can run a gel, electrophoresis gel, to get a pattern, and you can see if that pattern, based on that limited amount of DNA at the crime scene that you've multiplied with this PCR technique, now you can compare it to a DNA sample that a suspect may have been required to submit, and you can see, does the suspect profile fit the crime scene profile, and that's very powerful information for law enforcement to know whether they have the suspect who is likely the suspect that was present at the scene, or if that suspect is not a match based on the PCR and DNA testing. So in the PCR technique that we just discussed, we're taking DNA and multiplying it. We're not trying to change it. Indeed, we're trying to keep it the same. Another way that humans have gotten involved in the genetic code is through recombinant DNA. And that's DNA that's produced artificially and will contain DNA from two different sources. So in one method, you take restriction enzymes to cleave the DNA, and then you take those DNA fragments and insert them into plasmids, these sort of circular DNA things that bacteria have, and the plasmid gets inserted into a host organism, replicates itself, making copies of itself. And this is how we can do things like making goats that produce silk in their milk by taking the silkworm instructions and inserting it into the DNA of the goat. You now have the ability to have goats that express silk proteins in their milk, and obviously it's much more abundant source of silk than those tiny little silkworm cocoons that we've been relying on until recombinant DNA technology. And here we have a cartoon of that recombinant DNA where you take the plasmid from something like an E. coli bacterium and DNA from a human cell in this example, and the fragment is of the human DNA that has a particular protein we want, is cut with restriction enzymes and then inserted into the plasmid, and then those bioengineered plasmids get introduced into additional E. coli, and we can suddenly mass produce this desired human protein in these bacterial colonies that we've genetically modified through recombinant DNA technology. So one of the goals would be to have something like gene therapy, where you'd have a functional gene in a person who might have a defective gene. You can insert this functional gene into that person's genome, and now you've cured them of their genetic defect. Viruses can be used to carry the DNA into the cells, but again, this is still experimental, and it brings up a lot of ethical questions and concerns about what we're doing, and is it right to do this? Is it ethical to do this? Are we really at the state of the art that we can do this, or are we really moving too fast, and are there a lot of drawbacks that we might not have thought about that could be a consequence of moving into this realm without fully understanding all the consequences of our actions? So I suppose that's where we'll have to end this chapter. Hopefully a very interesting chapter, one that, again, in a single chapter, it's tough to give you a flavor of all that biochemistry holds, but hopefully at least you'll take home the message of the four major macromolecules: the carbohydrates, lipids, proteins, and nucleic acids, and that the more we learn, the more questions that arise, and certainly in the case here of genetic engineering, we've done a fair deal already. We're able to see that cloning animals and plants has a lot of potential promise for food production, disease treatment. However, there's also a lot of controversy and concern. If we make these genetically modified organisms, are they going to work well with a landscape of organisms that haven't undergone genetic modification? And so you see a lot of non-GMO foods, for instance, being marketed because there is some concern about the genetically modified organisms and whether or not they're safe, whether or not they're safe to consume as an individual, safe for the ecosystem in general. So lots and lots to discuss, and a lot of potential promise and potential concern that will be the case for the foreseeable future in genetic engineering. It's going to be a lot of controversy before we get much more progress. So you all will see more of that and reap more of the benefits and hear more about the controversy than I will. Hopefully you've enjoyed our brief foray into biochemistry, and maybe you'll consider taking a more advanced biochemistry course in the future.